Great to be here. First of all, thanks a lot for the invitation. So Elypta is a spin-out from Chalmers. It was founded just a bit more than 2 years ago. And let me get the clicker. We were founded with the vision to enable closer follow-up and earlier detection of cancer. So the problem that we see in cancer care today is that we maybe rely too much actually on medical imaging. There are some issues here. We expose the patient to radiation risk, at least with some modalities like computed tomography. It's also perhaps a challenging thing to detect really small tumors. You literally need to see it, right? So this can be an issue in motivating the use of this technology frequently to find the cancer earlier. So ideally, you would want some type of a molecular approach, right? You'd want something that's invasive, preferably, and something that's really sensitive. So a liquid biopsy. And there's a lot of companies and groups around the world that are focusing on trying to enable this for cancer. Our approach is differentiated in that it's based in metabolism. And I'll tell you more about that. So it's inherently safe and convenient. It's a urine test. We're also working on a blood test. We are different in that our technology seems to enable a very sensitive test also to small tumors or early stage cancer. And this we think is very important. Also, it can be made affordable at high volumes, and it's very, very much scalable. So all of this enable a test that you can actually use frequently and that you can motivate to set in at an early stage, ultimately enabling early detection of cancer. So metabolism, I mentioned. Why metabolism? Well, we see a lot of efforts, and rightly so, in genomics and proteomics to try to find biomarkers for cancer. And metabolism, we think, is another good source that we should look more into. It's sort of downstream of DNA and proteins. If you think of the DNA molecule, it's actually quite static, right? It's good at being predictive of risk or therapy response. But metabolites are very much dynamic, and they're very sensitive to biological change. If you think of it as a layperson, you can, and I'm a layperson, you can think of it as if you hold your breath, the metabolism will change. The DNA molecules hopefully will not. And this is something we want to utilize. So then the question is, what is it that we should measure? Well, this was part of the question that was asked at Chalmers in Jens Nielsen's systems biology lab, more specifically. And you could simplify the question as what should we measure in metabolism that we can use for perhaps diagnostic purposes of cancer? And it turns out that there's an exceptional regulation of glycosaminoglycans in cancer. What we see here is the glycosaminoglycan biosynthesis. So it's a lot of metabolites, you could see it as. In fact, there are 19 biomarkers that we've developed a way to measure in Elypta. And this pattern of metabolites is changing in cancer patients. And that's what we're tapping into. So Elypta has developed a way to, an assay, a laboratory assay, to measure these 19 metabolites, and then basically use machine learning to find the patterns specific to cancer. And it turns out it's different in different cancer indications. Now, as mentioned, what we can do is condense the 19 measurements into a scoring algorithm so that we get one single score. And that's what we see here. So here are two proof of concept studies. The first one on the left, actually done here, partly at Sahlgrenska, also in Italy. And you see on the y-axis the GAG score, if you will, the condensation of the 19 measurements. And you see plasma and urine samples, and the gray bar here is kidney cancer patients, so metastatic kidney cancer patients. And then you have a control group which is healthy. And as you can see, it's very clear separation here between healthy and cancer. In fact, it's even 100%. And in this discovery study. Then we moved on to test this also for early stage cancer. And that's what you see on the right here. So this was a study with MSKCC in New York. And the interesting part here is that we have a healthy control group, red dots, but then stage 1, 2, 3, 4 cancer. And as you can see quite clearly, we can separate also between the early stage and the late stage cancer. So this is powerful. Overall, it's more than 93% accurate to any stage kidney cancer in these studies. Now, this is, as you know, many of the scientists in the audience, it's a discovery study, proof of concept. The important thing is, of course, what do we use it for? Well, our first application is the follow-up of actual kidney cancer patients, so those that have already been diagnosed. About 90,000 have surgery in EU and US every year, but 20% of them get the cancer back. So it's important to be able to follow them. And today, this is done with computer tomography. So there are no biomarkers available in kidney cancers at all. And this is something we want to change. So there are 2 studies ongoing at Sahlgrenska right now. There are more actually, but 2 within kidney cancer and our biomarkers. And one of them asks the question, so for patients that have metastatic cancer and they're treated with therapy, can glycosaminoglycans or GAGs detect progressing cancer perhaps sooner? Can we find it in a good way, not just using CT? And then perhaps we'll have to see later on, could we switch therapy, adjust the dose, etc.? There are lots of different things an oncologist can think of doing. The second question, which we'll focus more on initially when we go to market, will be, we have to see depending on how all the data comes out, but can GAGs detect a relapsing cancer earlier? So this is for patients that had surgery, not yet on therapy, they're effectively treated, hopefully curatively, and the question is, can we see if it comes back? And if we do enable to do that, we might be able to for some patients, enable surgery again. And that's really the only curative option for these patients. So that would be a great impact that we're all hoping for. And we do this by taking a sample before surgery, and then we take measurements regularly, and we see if the GAG score, as we call it, go up. I can't show you the data on these studies yet, but they are very promising, and they've given us reason to now proceed with the largest study ever in kidney cancer diagnostics. It's called Aurora X87A, and the goal is just this, is to detect a recurring kidney cancer, hopefully earlier, and it will be 16 sites in 6 countries that will be involved in this, 280 patients. And we got a grant from Horizon 2020 to do this study. So this, we hope, will make a big impact. And the objective is of course, approval in Europe, but also from the FDA to go to the US market. So what's our product? Well, first is a kit to measure the glycosaminoglycans. This is what we spent a lot of time developing, an extraction kit to measure the fold profile, but then we also need a software to be able to interpret these measurements and develop the score. So we're developing that in the cloud, of course, and it's possible to add more algorithms for other cancers or other combinations over time. It also connects, or is capable of connecting to the EMRs. And we think this is very important, partly to deliver the results fast back to the clinician and ultimately the patient, but also to be able to extract data with the patient's consent, of course, back. So perhaps we'd be very interested in understanding what the eventual diagnosis was. So can we continue to train the algorithms with real-world data? That's what we want to do. We also want to do combining diagnostics, not companion diagnostics, but combining that. So combine this with the PSA test, for instance. And use the power of bioinformatics. So there's a small but growing team at Elypta dedicated to doing this. Francesco is, of course, worth mentioning. He's the inventor. from Chalmers, and now full-time CSO in the company. He's working together with Nils on the algorithms and the systems biology around this and the software development. Karin is our new VP of R&D. She's working together with Swad on the assay development, so developing on the laboratory side. Mariana is running our big clinical trial, which is really a full-time job to have 16 hospitals. And of course, to mention also the customer, here represented by Saeed Abustani, who's a urological surgeon and an advisor to Elypta. There are many suppliers also involved in this project, and several of them here in Gothenburg, and really development partners to us. So it's a big team when you think of it. It's been a great year for us. With lots of recognition, and we think partly because it really sparks people's thinking with the possibility of early detection of cancer. So we feel that mission hits a trigger with a lot of people. And what we've done couldn't have been done without the backers that we have, many of them coming from here, of course. Sahlgrenska, but also Chalmers Ventures, etc., that has funded the original research and now continues to fund Elypta going forward. So, yeah, that's great. Thanks, everyone, for the attention.
Great. Thank you. Yeah, it's quite impressive what you have done.
Thank you.
In such a short amount of time here. And I know that you just yesterday arrived from Boston.
Yes.
And was nominated. I don't know, I didn't see it from there. Was it up there? You were nominated for, like, it's a MedTech Oscar. Yeah?
Yeah.
Yeah?
That's what they want to call it, at least.
Yeah? Yeah, sure,
yeah.
Of course.
Yeah, so we travel a lot. Unfortunately, we need to work on the commercialization side. So that's a big part of it. We want to go to the US markets, and we're talking to diagnostic companies, but also pharma companies and laboratory companies, of course, to be able to deliver this test to market. So
yeah, like you said early on in your presentation, it's a test that you can be frequently used. What do you mean by that?
Well, it's simply if you have a test that is safe and non-invasive and that has a clinical use, it has a clinical utility. We believe that for some indications it's a purpose to be able to use the test often, right? More often than we do today, to then find the cancer when it's coming back. You don't know when this will happen. Sometimes the follow-up is every 2 years. You need to be lucky as a patient to have the cancer come back right before the follow-up. So this is what we want to add, another tool.
Okay, so that's maybe the idea behind combining it with, like, PSA tests and others that you mentioned.
It's a possibility. So yeah. we're looking at different cancers as well, of course. So kidney cancer is our first priority. That's what we want to go to market with. But we're also doing studies within prostate cancer, and here at Sahlgrenska, bladder cancer. And when I say this, it's really academically sponsored studies. The Elypta's own sponsored study will be this big go-to-market trial for kidney cancer.
Yeah, I work quite internationally already. You were mentioning this study coming up with 16 sites.
Yeah.
So where, in what countries? Are they all in Sweden, or?
It's the US, and then it's 5 countries in Europe. So we haven't signed the contracts with the sites yet. So I can't really announce them yet.
No, but it's international.
Yes. Yeah. It's 5 European countries and 3 sites in the US at least.
Yeah, and this also shows that life science is global from start, which differs this sector from many other sectors, actually, because the challenges are global, so to say. Definitely. And you were mentioning there the other areas, and I mean, you were looking out for other tests to combine with. So this calls for collaborations. How do you work with collaborations?
Well, for one, the most important thing for us now is the sort of scientific and clinical collaborations to gather the samples to do the clinical studies, right? But as we move forward, it will also be collaborations with the pharma companies, for instance. It's very, very interesting. So the idea can be to bundle a test or an algorithm, really, with our kit to monitor the response to a certain drug. That's one possibility. Or if we're lucky and the only way to find out is to do research together, is to figure out if our test in any way can help predict what drugs work better than others, right? So these are typical research topics that we'd want to explore.
Well, thank you, Karl.
Thank you.
A big hand.